much. Thank you, Vijay, for that very generous introduction. And it was so touching to hear about Dr. Kirpal Chok, the way you described. I think you captured this not only his exceptional expertise as we all recognize as father of nephrology in India. We actually recognize him as a father of nephrology internationally. And also you captured this beautiful humanitarian spirit. It is, I think we have all been so fortunate that we had somebody like Dr. Kirpal Chok to kind of mentor all of us and lead the way. And of all the privileges and honors I've ever received, I cherish giving the Kirpal Chok oration in 2001, as Dr. Bala mentioned. I do get invited to different conferences, but of all the conferences I go to, India conference is my most favorite one because I'm back home and I have the distinct pleasure of interacting with wonderful friends like Vijay and others. So thank you so much for this high honor and privilege to give the Dr. Chok Memorial Oration. I wish it was not a memorial, just was an oration, and to participate in the Avatar Conference. Dr. Ajay Kar sent me this title. Since I'm a very obedient person, I didn't change the title, I stayed with it. So this is, I'm going to speak about current state of immune tolerance in kidney transplantation, the Holy Grail. This is clearly the much cherished goal. I'm very pleased to share with you some of the information. I think it's important to define tolerance. We want to talk about some mechanisms, not obviously all of them. And I will show you a preclinical transplantation model. There have been many studies, but I've chosen to present data from my lab because I understand my data fairly well. It's not that very easy to understand all that is published in literature. Then I'm going to discuss some clinical tolerance trials and also give you some introduction into biomarkers of tolerance, which is kind of the counterpart for biomarkers of rejection, with which we have been working for almost 2 decades. To start with definition, in most textbooks of immunology or transplantation, the conventional definition is absence of allograft-directed immune response without immunosuppressive therapy. This is very good. This has been the prevailing definition for a long time. An absence of an immune response directed at the allograft without exogenous immunosuppressive therapy. I've been in a number of tolerance conferences, and I was kind of not fully happy about this definition. So my definition of tolerance is absence of allograft-directed destructive immune response without immunosuppressive therapy. I think the word destructive, the absence of a destructive, is important because this new definition allows for allograft-directed immune response that is graft protective. It's not simply an absence of immune response. There is an absence of a graft destructive response while there is the presence of an graft protective response, such as regulatory T cells. And in the past, we used to call it suppressor cells. I think this is an important distinction about the conventional definition, how we may want to think about tolerance definition as we go forward. What about tolerance mechanism? Tolerance mechanism is extraordinarily important because without tolerance mechanism, we would hurt ourselves. Type 1 diabetes, systemic lupus. Think about all the autoimmune diseases. These are prevented by tolerance mechanism because the immune system has to attack the invader, but not attack ourselves. And there are at least 3 basic mechanisms for tolerance. One mechanism is that this is what happens in vivo. We all do this. Otherwise, we would have many, many difficulties like type 1 diabetes and lupus. One mechanism is called clonal deletion. There is a physical absence of antigen reactive cell. The immune response is very specific. If you don't have antigen reactive cell, that for that appropriate antigen, is the reactive cell is not present, that's clonal deletion. There is also a possibility that we will have cells that are capable of reacting but do not react. They're functionally inert, and this process is known as clonal anergy. The third mechanism is a suppressor mechanism. The cells are present, they can act, but they are prevented from acting by extraneous forces like cells, anti-idiotypic antibodies, or T-regulatory cells. These are the three fundamental mechanisms by which we protect ourselves from autoimmune or a self-destructive immune response. And these are the very mechanisms that operate to create tolerance. So when people want to create tolerance, we are kind of leveraging one or other of these mechanisms. When I teach at Cornell, I kind of use this example for the medical students. I tell them clonal deletion, physical absence is something like you never showed up for this lecture, you're physically absent. Clonal anergy is you showed up for the lecture, but you never listened to anything I said. You're physically present, but functionally inert. Suppressor mechanism is you're here for the lecture, you actually like it and you're trying to follow the lecture, but people around you are preventing you from learning. So these are the three mechanisms. The Cornell students like these examples. We all as physicians receive so much of information. Maybe if you use that analogy, you will have a good idea about tolerance mechanisms. One of the fundamental thing about T cell activation, and we need to know this in order to understand tolerance. T cells, you all know as a T cell receptor, it is connected to the CD3 complex protein, which is gamma, delta, epsilon, and 2 copies of zeta chain. The T cells are very formal. They don't simply see the antigen. The antigen has to be presented by antigen presenting cell like a dendritic cells or macrophages or B cells. When the antigen presenting cell presents the antigenic peptide, there are two signals. One signal is the T cell activation signal to the TCR CD3. A second signal, a co-stimulatory signal, and these two signals together results in T cell activation. This is a very key concept. The T cells requiring two signals, the antigenic signal and the co-stimulatory signal. We emphasize these two components because if you block the co-stimulatory signal and let the antigenic signal go through, you get what is known as an anergy. And this is one of the mechanisms, when you use CTLA-4 Ig fusion protein or a belatacept in your transplant patient, this is what you're trying to block, that interaction between CD80 and CD86 and the CTLA-4 and CD28 on this T cell. So blockade of co-stimulation and creating anergy is one important mechanism for tolerance. And we have done this, and many others have done that. In fact, we are not the first one to do it. Bluestone and others showed it in a mouse model. But I would like to show you what we did because that has direct implication for clinical care. The model we use is a mouse islet cell transplant model. In the mouse, you take a normal mouse and you treat the mouse with STZ. This is an islet toxin, and the mouse become diabetic. Then you isolate the islets, and then you place the islet under the renal capsule. The reason you put the islet not in the pancreas, but in the renal capsule is because the renal capsule is highly vascularized. It's not because we are nephrologists and we got confused and put it under the renal capsule. Here are the islets that were isolated in my laboratory, which is a fairly complex process to isolate islets. I was very fortunate to have Dr. Hua Yang in my laboratory, who was one of the specialists in islet isolation. This is a mouse islet. We also went on to isolate human islets and perform successful human islet cell transplantation. But the data I'm going to show you is from the mouse model. So we took these islets from a B6A1 mice, and we gave this antibody. This is what is the survival time following transplantation. The survival here is monitored by blood glucose. In a diabetic mouse, if the glucose is normal, then the islet is working. If the mouse becomes hyperglycemic, the islet is rejected. So when we gave an antibody called anti-CD2, we gave CD2 because many years before in my laboratory, we identified CD2 as a very potent co-stimulatory molecule. In fact, when you give your patients thymoglobulin, and I know many of us use thymoglobulin as an induction reagent, the most important component of the polyclonal thymoglobulin may very well be anti-CD2. So 21 mice were treated with this, and 19 mice were not treated with it. And as you can see that, most of them lost their islet cell graft. There was always one or two. This is why it's important not to just do 3 mice or 6 mice. You have to do a large number of experiments. Here we have done 40 mice experiment. There's always one or two who kind of keep the graft without any treatment, not unlike our clinical situations. And here, when you give anti-CD2, I don't have 100% survival, but I have a significantly better survival. So what this slide shows is that if you block the CD2 co-stimulatory signal with a monoclonal antibody directed against CD2 antigen, you are able to prolong survival. This doesn't mean there is tolerance. Tolerance has to be antigen specific. So in this experiment, what we have done is a perfectly functioning islet. We take the kidney out. We take the kidney and islet out, and then come back with either a third-party islet or the same party. This is the original donor was DBA2 mouse. The third-party mouse was an SJL. And you can see a very striking impact of how brilliant our immune system is. When you come back with the third-party mouse, now this looks like it's not been tolerized, so they promptly reject these islets. Whereas you come back with the islet you previously transplanted, DBA2, you get a prolonged survival without any new treatment at all. That is, the immune system has been educated to tolerate this graft. This is a hallmark of a tolerance. That is not just lack of immune response, there is an antigen-specific lack of immune response. This was done by Sandy Kapoor when he was a surgical resident at Cornell. Now he's our chair of our transplant surgery. And we are very pleased that we were able to bring up transplant surgeons, not just only nephrologists and transplant physicians in our laboratory. Now, what about clinical trials? In the interest of time, I'm just going to focus on few clinical trials. I apologize to those trials that I've not talked about. India has reported from Ahmedabad and other places. There has been a number of studies. I have selected clinical trials in which we participated. We participated primarily from doing immune monitoring. I think one of the important concepts I would like to introduce before we go to tolerance is this idea of chimerism. Chimerism defines, let's go here. Chimerism is a state in which hematopoietic cells are present in the recipient organ, donor hematopoietic cells. So you're a hybrid. I think we have some Indian guards who are hybrid. one half is male and one half is female. It's probably one of the most classic example of chimerism. But chimerism seems to be important, that you have to either infuse bone marrow itself, or you infuse hematopoietic stem cell like CD34 cells. And there are different types of chimerism, mixed chimerism, where you have recipient and donor, full chimerism, where completely donor cell. The chimerism can be stable, or durable, or transient. And here is a flower I kind of got from where we all have a source of knowledge, internet, showing a chimeric flower. Now, this is important concept, chimerism. And the first study, this is one of the first studies of chimerism developed by the MGH group, Mass General group, led by David Sachs. I had the privilege of being a co-editor with them on the journal Transplantation. This whole idea came from David Sachs and his team. I'm showing this, not that this is the only way to achieve tolerance, but to show this is fairly an intense protocol. You pre-treat the patient, then on the day of transplantation, you give a kidney and a bone marrow, and then you give this anti-CD2. This is the monoclonal antibody or the antigen we had identified as a good molecule to stop the immune response. So there is a preconditioning, there is transplantation, there is a kidney and a bone marrow transplant, and then you give immunosuppression for a duration, maybe 6 months, 12 months, and then you look for chimerism, and then you stop. As you can see in this slide, these are 3 protocols that David developed. One is called NKDO3, then it gets modified, and then an immunotolerance protocol. Just to make the point, we don't have one protocol that's perfect. We are still learning, and we are kind of generating different protocols. So it's not necessary for you to precisely know this protocol, but it's important to know it's a fairly intensive preconditioning, transplantation, bone marrow, some immunosuppression for a period of time. This was published in the New England Journal of Medicine in 2008. This is the first example of a tolerance in an HLA mismatched renal transplantation. And the data has been updated recently by Kawai, who was the lead author. They have done now 10 transplants, all of them one haplotype matched. 4 recipients are 4.5 years to 11 years of immunosuppression. 3 recipients needed to be reintroduced. Drugs have to be reintroduced either for recurrence or rejection. This is another very interesting thing. The primary kidney disease, even if you're tolerant to the transplant, the primary kidney disease can come back. And unfortunately, 3 grafts failed. And the failure looked like thrombotic microangiopathy. This is like, we had a beautiful lecture about complement and ABO incompatibility resulting maybe in a TMA-like picture, or it could be an antibody-mediated rejection was not clear. So 10 patients have been done, 3 failed, and you may want to worry about it. But there are, for patients who are off all immunosuppression for 10 years, and in the next 6 months or so, I think this trial will be reinitiated at Columbia, where David is, and Cornell and Harvard in the next few years. And we are learning. We're still learning. It's not like it has all been solved. And then in these patients, the kidney biopsy sample was sent to my laboratory, blinded, and we did gene expression profiling. This is what we've been doing for 20 plus years. We developed a fine type of a polymerase chain reaction, a quantitative PCR. And what this shows is a FOXP3. Why FOXP3? FOXP3 codes for regulatory T cells. And I told you about lack of a graft destructive response, but maybe a presence of a graft protective response. And this shows that FOXP3 in this tolerant patient is significant. This is actually, the data is a 18S ratio. This is actually very significant that the patients who are tolerant have a much higher FOXP3 than patients who are on immunosuppression and normal. Very interestingly, even during acute rejection, FOXP3 goes up, and that may be very, very protective. Unlike FOXP3, granzyme B, which codes for a cytotoxic T cell, it's really, the stable patient and the tolerant patient and the stable patient is very similar. So altogether, what this data shows that during tolerance, patients have a graft protective immune response exemplified by overexpression of FOXP3 and lack of a graft destructive response exemplified by a low granzyme B. So that's the Harvard study from David Sachs' team. And Dr. Strober has been a pioneer from Stanford. He developed a beautiful protocol of tolerance and chimerism after renal and kidney transplantation. This was published first in 2008. This is a single patient. As you know, it's very difficult to publish in NEJM. And this was a single patient which got into NEJM because this was so groundbreaking. But this was an HLA identical transplant, the first one. They used thymoglobulin, which is anti-CD2, total lymphoid irradiation. When we say hematopoietic stem cell, we mean CD34. They also gave CD3 T cells. They were treated with, first patient was treated with cyclosporin, later patient with tacrolimus, PRED, and our standard immunosuppression, which was discontinued after 6 or 12 months, persistent chimerism. What does it mean? The presence of donor cells in the recipient, no rejection, no GVHD. Now they have done 29 HLA-2 haplotype matched patients. 23 of 29 are rejection-free for 22 years. This is probably one of the largest induced tolerance group. Unfortunately, if it's that one haplotype matched living donor grafts, they had to keep them on monotherapy. So they're not really tolerant in the classical sense. But I think if you can minimize immunosuppression, I think that's a very, very good goal for us. In fact, I like minimization of immunosuppression very much. And I think that's more an attainable goal than a complete stoppage of all immunosuppression. Clearly, you will have some patients who will be fine, and there will be some patients who can get into trouble, just like here in the update they published recently, 2 patients lost their grafts. The third group that has done a beautiful work with tolerance is the group of Joe Leventhal from Northwestern and Susan Ilstead. Susan Ilstead developed this a very interesting cell called facilitator cell, and they published this data in Science Translational Medicine in 2020. And this is also a complicated protocol, cyclophosphamide irradiation. I think you get the idea that you need really preconditioning. You need kidney transplantation, you need bone marrow plus hematopoietic stem cell. They treated this patient with tacrolimus and mycophenolate mofetil. And recently the data has been updated, and this has not been published yet, but they have about 37 patients, 26 of 37 with durable chimerism and off immunosuppression. There's a 94% survival rate and less than 3% at a severe GVHD. This is a total chimerism, this model, and graft-versus-host disease is a real problem in this situation, as with any tolerance model. And we as clinicians need to think about whether this is a price worth paying, graft-versus-host disease, whether that's a good price to pay to have patients of immunosuppression. These patients' urine samples were sent to my laboratory, and we profiled a variety of genes. These are Fox and Bucks and Whisker plot, and we looked at a gene called CTLA4. This is a negative regulatory molecule. Very interestingly, the tolerance patient have a significantly higher CTLA4. No rejection patients from a CTAT-04 study we were involved in don't have it. Even ACR patients don't have it. This is very, very unique, suggesting upregulation of CTLA4 may be very important for tolerance. And you can see granzyme B is low, and the CTLA4, a protective immune response, granzyme B, a destructive immune response, is clearly tilted towards intolerance patient. This is what I like the definition A tolerance is an absence of a graft-destructive immune response and allowing for the presence of graft-protective response. There are many other trials. I have not captured every one of them, including some very interesting studies from India. There was a study about the HLA identical patients from Ahmedabad, but on a monotherapy on some time, but there could be many things I might have missed. I just wanted to present some select cases given the time constraint and also the fact that we really studied these patients ourselves. So we are very comfortable about the data. I think when you talk about tolerance, you cannot help showing this slide. This is one of my most favorite slides. I think I might have shown it when I gave the Dr. Chuk oration in 2004. Tolerance in our time. I think this is very important. I think there's a beautiful talk about the medical ethics about treating people well. I think we have Mahatma Gandhi taught us about tolerance, not just between people and between religions and between countries. And we are so fortunate to have, this kind of a cultural background that we think about tolerance. I do think tolerance is doable in the clinic. It has been done. We have participated in a number of pioneering studies. Those models came from Stanford. Those models came from Harvard. Those models came from Northwestern. And we were fortunate to do some profiling studies. But I think minimization of immunosuppression is an important goal as well. So I thank you again for this high honor. It's always a privilege. It's always a pleasure for me to participate in a conference in India. And I wish the next time we will meet in person. This is my last slide, and I would be happy to